Good morning, everyone, and I welcome you all on our day 2 of our global initiative of academic networks GIAN course. To begin with, today we have our distinguished guest, Indian expert, Dr. Rajesh Badani. It is my honor and privilege to introduce a highly accomplished clinician, teacher, and researcher in the field of cardiology, Dr. Rajesh Badani. He is a distinguished senior consultant, interventional cardiologist, and electrophysiologist at Aditya Birla Memorial Hospital, Pune, where he also serves as a cath lab coordinator, administrative in charge, and patient safety officer. He completed his MBBS from SET GS Medical College, Mumbai, followed by a MD Medicine from Grant Medical College and DNB Medicine in Cardiology from the National Board of Examinations, Delhi. He further specialized with a fellowship in electrophysiology at Care Hospitals, Hyderabad. Dr. Badani has over 10,000 diagnostic catheterizations, 2,000 plus coronary interventions, including complex CTO and bifurcation angioplasties, more than 2,000 pacemaker implantations, along with extensive experience in AICD, CRT, and valvotomy procedures. His expertise also covers a wide range of non-invasive cardiology diagnostics, including echocardiography, Holter test, and tilt test. He has held key positions in professional bodies, namely President from 2012 to 13 and Secretary from 2009 to 10 of the Cardiology Society of India, Pune chapter, and is a fellow of the European Society of Cardiology and FSCAI. His notable research contributions include Brugada syndrome, atrial tachycardia, electrophysiology case studies, mutations in myosin 7 and troponin I3 genes in hypertrophic cardiomyopathy, oxidative stress in cardiovascular disorders, diabetes, and anemia. He has a strong research portfolio, having authored numerous peer-reviewed articles and conference abstracts, including work published in top-tier journals such as the Journal of the American College of Cardiology and also the Indian Heart Journal. His research span areas from electrophysiology and pacing to genetic studies in hypertrophic cardiomyopathy. He is also the recipient of the D.P. Basu Award for the best paper at Cardiology Society of India in 2008 and continues to contribute actively as an invited faculty at national and international conferences. Through his blend in clinical expertise, leadership roles, and impactful research, Dr. Badani has made significant contributions to the advancement of cardiology in India and beyond. Now, I request our course coordinator and our HOD of Department of Zoology, Professor Narahari Grampurohit sir, to welcome and felicitate Dr. Rajesh Badani. Thank you, sir, and thank you, ma'am. Now, we begin today with our Lecture 3 of Day 2 course, Diagnostic and Genetic Aspects of Hypertrophic Cardiomyopathy by Dr. Rajesh Badani. Sir, please come and grace us.
I'm okay with this also. Color mic, yeah, it will be better. Hello. Yeah, pretty good. Presentation. Okay, good morning all. First of all, I should thank thanks. Dr. Varsha ma'am, as well as the Department of Zoology for having invited me here. I think our association almost dates more than around 8-10 years back, when we probably started doing brief genetic work in hypertrophic cardiomyopathy. And I believe it's great to see that the department is really doing very good work in basic sciences, genetics, and particularly focus on the hypertrophic cardiomyopathy. And this program, when I saw the agenda, I think it covers almost every single aspect. Today's talk that is going to be is on genetics in hypertrophic cardiomyopathy. What are the clinical implications for the diagnosis and management? So my key discussion will remain to genetics, which probably you guys and I think Professor Sakthivel is here, so maybe apologies if I am not an expert. That is the thing in genetics. Though whatever is there, it's more of a clinical association. Little bit of terms I may still not be really very much aware, which I think will take the experts' help. So hypertrophic cardiomyopathy is the prototype genetic cardiomyopathy. Almost 60 to 70% of the people with hypertrophic have got an association of genetic association. It is there. It runs in family, predominantly an autosomal dominant condition. So that means that the offspring has a 50% chance of acquiring the gene from the parents. Now the only thing is that the moment the person has a gene which is a genotype positive, phenotypically how the patient will behave, that is the most interesting variability. Many people don't present anything till 3rd to 5th decade. Some people manifest very early in childhood, some people in middle age, but average is 3rd to the 5th decade. So that is between 25 years to 45 years is the time when the diagnosis, symptoms and other things come up. Now understanding the genetic background can enhance our insight into its pathogenesis, number one, and the potent therapeutic targets. While the clinical presentation is variable, remember why it's variable, because you can have a person who is absolutely asymptomatic versus the person can present with sudden cardiac death. And advances in molecular genetics have really revolutionized. In US, I think from where Professor hails, they have a very, very dedicated way of evaluating hypertrophic cardiomyopathy from animal to humans. They have got a great genome project. They have got multiple genes which have been identified, but in India we are still at the inception stage. Now this is very, very interesting to understand that the etiology of hypertrophic cardiomyopathy can be considered infective, hereditary in 70%, hypoxia and oxygen imbalance has been considered one of the etiological processes. Then comes the hormonal imbalance. Many people in presentation, now these on the left hand side are the etiology, this is the presentation. Majority are asymptomatic. Somebody can have a ventricular tachycardia. Atrial fibrillation is also very common. They can have thromboembolic episodes and stroke which can occur because of AF. And the most interesting aspect is the commonest cause of sudden cardiac death in people below the age of 35 years is nothing but a hypertrophic cardiomyopathy. People who have had, you have seen soccer players who probably have fallen on the ground, not woken up, or somebody was defibrillated and they got alive, majority. So these were undetected HCMs going on, and many of them can be sporadic. So they may not have a family history. The person index case may present with hypertrophy, syncope, resuscitated cardiac arrest, and that's when you diagnose this patient. That is why this condition becomes very important from genetics to the clinical presentation to be identified. So hypertrophic cardiomyopathy is an abnormality of sarcomeric gene proteins which are there predominantly. Non-sarcomere and mitochondrial account for less than 20% or 5% or 20% of the total number of genetic which is there. So if you want to understand, this is the sarcomere, where these are the thick filaments which are blue, the green are the thin filaments, or the thick are called obviously myosin, thin are called actin, and the actin myosin complexes, when they interact, that's when the excitation contraction coupling occurs, a heart contracts, and probably we see the contractile phase. So that is how at cellular level these are the things. There is something called as a M-line protein, and these are calcium related genes. So commonest are the thick filaments. Almost majority, more than 40% of genes come from this, particularly the myosin heavy chain 7 or myosin binding protein C3. These are the two names that you should remember very clearly. Majority of HCM is because of these two genetic abnormalities. Thin filaments will come to troponin T, troponin I, troponin C, and you have myosin light chain 2 and 3, which are obviously also. So these will account. So total of 5 to 7 and to 9. 8 out of these 9 genes are definitively associated with hypertrophic cardiomyopathy. All others may not have a very strong association, which are these. This is titin, Z disc proteins, as well as the calcium and the M line. These are all very rare. Z disc, particularly CRSP3, is very strongly associated with sudden cardiac death, and now we have therapy, gene therapy, which is probably being working on that. I will come to it later. Now, how does an abnormality in the genetic association cause an abnormal phenotype presentation of hypertrophy and hypercontractility? So for all of you who have been there yesterday in Dr. Chauhan's talk, it is very clear that in hypertrophic cardiomyopathy, you have higher contraction which is there. Why do you have higher contraction? Actin and myosin are strongly associated, and when they both get in contact with each other and there is an ADP-ATP conversion, that is when the contractility improves. In the upper panel, just for simplistic purposes, these are the only two myosin is in touch with actin. The red and the green are in touch with each other only in 2 of these. Here, this is the HCM heart 1, 2, 3, 4. So when you have a larger component of actin myosin contraction, the chances of higher contractility becomes more. At the same time, the relaxation phase, which is the diastole, gets affected, and that is the reason hypertrophic or any heart which is thick does not relax very well. It contracts excellently, but the relaxation is very slower. That those on echo and other features we identify, but from this perspective we need to understand this is the basic pathophysiology, how does it occur. Now location within the cell, if you take sarcomeric or the contractile protein, Z, look at the incidence, 40 to 50%, 35 to 40%, 7 to 15 is troponin T, troponin I is 3%, myosin light chain is 1 to 2%, all others are below 1%. So that's why I am telling you that if you are able to identify these 7 or 8 gene, and that's why the gene panel, when we ask for a genetic testing, takes care of all these things, because your majority of cases are going to be there. Calcium binding protein, it has a definitive evidence, and it is PLN phospholamban. It has got a specific cardiomyopathy which we call it as phospholamban related cardiomyopathy, but that is very rare. And there is only one gene which is probably X linked. Where it is gone, right? Here it is. LIM domain protein, which is a Z disc protein, which is FHL1, and it is probably the only one which is an X linked. All others are autosomal dominant, and few of them, the uncommon ones lower down, are autosomal recessive. So your predominant understanding should be it's an autosomal dominant. Family history has to be very strong. There is a 50% chance, and few of them can present with sporadic cases. They may not be having any past history. Now list of the main genotype, when you say, these are called as hypertrophic cardiomyopathy mimics. On echo, I have a thick septum and a posterior wall. So every thick septum does not mean it is hypertrophic cardiomyopathy. It could be one of these: metabolic regulatory, lysosomal or glycogen storage, lysosome, RAS MAPK pathway, or others like transthyretin, which we call it as cardiac amyloidosis, and mitochondrial diseases which are there. All of them are definitive. Lysosomal as something called as LAMP2, and PRKAG2 is the commonest two, which is called as PRKAG2 syndrome, definitive. The Danon disease is X-linked. Anderson-Fabry, which is seen, these are all in young children. So any child you see with a very hypertrophied ventricle, you straight away think of this, do their echo, do their ECG, ask for family history, get your genetic analysis, and that's when you can prognosticate whether this child has a high risk, low risk, intermediate risk of sudden death, or future perspectives what you need to do. Now this is something, it's a busy slide, but this is just to understand that when we talk of genetic variants in the main genome, when we talk about, the slide is busy, but this is single nucleotide variations which we talk about. That is the most common type of abnormality, and then we talk about the other, which is uncommon with hypertrophic. So I'll skip this for the purpose of discussion, but you have to understand what is the abnormality. There is something called as an intron insertion or deletion, then you have mutation, then you have a lot of other terminologies which I am not very conversed with, but this is the commonest, single nucleotide variability. That is one of the commonest things that we need to know. Now categorization of genetic variant for clinical application. So when I see a genetic report as a clinician, what am I going to look at? These terms need to be, there is something called as a pathological variant, PV, or there is LPV, likely pathologic variant. Variants for which there is a strong genetic evidence for their involvement in the pathogenesis. So that means in the genetic report, if it comes as a PV, that means this gene has got a very strong association. So genetically this patient is genopositive. Clinically, as a doctor, I have to look whether he has hypertrophic cardiomyopathy. I'll ask for family history of death. I'll ask for syncope. I will ask for symptoms, dyspnea, fatigue, tiredness, all those things. See, PVs are predicted to distort the structure and function of the encoded proteins, often leading to premature truncation of protein and haploinsufficiency. These are called as frame shift variants, such as indels means deletion and insertion, and splice junction variants. Variants leading to gain or loss of a stopped codon. There is a CAD score which is called as a combined annotated dependent depletion score. It is very rare in the general population because means sporadic cases are rare, but familially this one is a very strong association. LPVs have characteristics similar to this. Typically they involve genes that are partially intolerant to LOF, is loss of function variants. Then you come to function variants. This is very important. A genetic report may tell you variants with unknown significant, VUS. So means that is not very strongly genetically associated. And if you see here, undefined functional or clinical significance, common in the genome but often located in intergenic regions or introns. There is something called as a likely benign and benign variants. These are common in the general population. Means in the population there are many people who will have hypertrophy. Athlete's heart and hypertension are two common conditions in the common population that you will see. So they will have this variety, but genetically they don't have PV or LPVs. So that's why their geno-pheno correlation is not strong. They will be genotype negative, but phenotypically they may behave like hypertrophy. Their prognosis is much better than the genopositive, phenopositive individuals. Now this is exactly the nutshell of the last two slides. On the left hand side, remember, this is population frequency of the genetic variants. Hypertrophy in response to external stimuli. Hypertension will cause pressure overload on the ventricle. which will increase the septal thickness, and athlete heart, you will have physiological hypertrophy because of excessive exercise. So, any external stimulus causes hypertrophy. It does not have a genetic correlation, but it has a population may the number is much more, number of pathological variants are numerous, but penetrance genetically is very low, and evidence for causality is only association. It is not a strong association like here. If I talk here monogenic and oligogenic hypertrophic cardiomyopathy, the effect size of gene variant is very high, but population frequency you will find is very less. They have pathological variants is only one. That means I told you like myosin binding protein C3 or heavy protein is present, but penetrance is very high. It can directly go across families. That is why the familial transmission is high and causality is very robust. Here it is moderate, weak, and only association. So, this is to differentiate a genotypic versus a phenotypic presentation, and how you will intermingle with these two things, and how I am going to do, how as a clinician we are going to evaluate. This is very, very important. This is also a complex side in the pathogenesis of hypertrophic. So, that is why this condition is very interesting, because the person may just be asymptomatic, but if I pick up that this hypertrophy is pathological, and if the person tells me that he has a strong family history of sudden death, and I try to get a gene analysis, there are certain genes which are directly proportional to history of sudden death, but on echo the hypertrophy is very less. That is what is very important to understand. So, see here at mRNA level, it is a stable expression or transcription efficiency or non-stop decay or no-go decay, mRNA stability and chromatin structure. These are the pathologic variants. Now, what happens at protein level? You have stable expression, that is I think what Dr. Sakthivel was talking about, poison peptide, translation efficiency. So, what does it do in the sarcomere assembly? It can be efficient or inefficient ATPase activity, actin, myosin. So, what happens from the genetic level, what happens to protein, from protein what happens to the cellular level of sarcomere, from there at cell level what happens, myocardial level what happens. Histopathologically there are three abnormalities. Number one is myocyte hypertrophy, myocyte disarray and interstitial fibrosis. So, if somebody only has hypertrophy, he will on echo behave as cardiac hypertrophy or LV outflow tract obstruction. He can present with heart failure with preserved ejection, he can come with breathlessness, edema, feet and all that. He can have ventricular tachycardia or atrial fibrillation and sudden cardiac death. So, this is the variability from genotype abnormality to the phenotype presentation. This is what happens, and it is such a mixed interplay that it is not 1 + 1 2, it is completely different. That is why this condition is very interesting, and you need to as a clinician suspect all these things and try to evaluate. So, what is the aim is to synthesize the current evidence what we discussed into the genetic architecture of HCM, including the sarcomeric and non-sarcomeric variants, inheritance pattern, autosomal dominant, autosomal recessive, X-linked or sporadic, genotype, phenotype correlation and implications for diagnosis, prognosis and treatment, which I will come to it at the end of the thing. Now, this is very important, role of genetic testing and variant interpretation. So, genetic testing is recommended definitely for index cases and at risk relatives to enable early detection and surveillance. High throughput panels, whole genome sequencing enhances detection rate to identify structural and deep intronic variants, improving the diagnostic yield to 12% as compared to conventional panels. Now, interpretation, I was telling you, you will have something called VUS, variants of unknown significance, remains a challenge, and all these guidelines help to optimize the classification accuracy and the clinical utility. How am I going to use this report and how am I going to treat the patient is different. Now, clinical utility of genetic testing is facilitates diagnosis in atypical cases, guides cascade testing in families, supports reproductive. A pregnant lady is there, what are you going to make a decision? You want to continue pregnancy, abort the child, what care are you going to take, all those things. Now, what does it do genetic? It will help differentiate thickened myocardium such as phenocopy diseases like amyloid, sarcoid, Fabry's, hypertensive, athlete's heart, and it guides the family surveillance. For pregnancy planning during genetic counseling, it should be clearly communicated to the proband and their partner about the inheritance. It's autosomal dominant. I have to tell that there is a 50% chance in the offspring that your child may carry this gene, and the potential for phenotypic development. Tomorrow I am transmitting the gene. Which gene will have a better penetrance? Also, I should be able to talk to the family, and then only the family can take a call of continuation of pregnancy or not to. Now, genotype phenotype correlation, mind you, is one of the most difficult situations. A person who is genotype positive, phenotype he can be negative, intermediate or positive. That is how. So, what certain mutations correlate with distinct phenotypes like myosin heavy chain 7 tend to produce earlier and more severe hypertrophy. So, they will present early, hypertrophy will be significant. Significant means more than 20 millimeter of septal thickness is called severe hypertrophy, and that is directly linked to increased risk of sudden cardiac death. MYBPC3, that is myosin binding protein 3, are associated with late onset disease and may exhibit incomplete penetrance. So, phenotypically the features may not be very classical because they are presenting late, third, fourth decade. So, it will be very difficult at a 45-year-old person who only has hypertension and hypertrophy, whether he has features of HCM. Young people, it's very easy to pick up. Why? Because I am supposed to be talking about SCD, ventricular arrhythmia. Should I put in an ICD in that patient or no? This is very, very interesting. TN and T2, they are often linked to a high risk of SCD despite minimal hypertrophy. A person may only have 12 or 13 millimeter septal thickness, but the risk of SCD, if he is of this genetic variability, is very high. So, if I have this genetic variation, I will straight away discuss with the family that I need to put in a device which is called implantable cardioverter defibrillator to reduce the risk of sudden cardiac death. Now, genetic contributions to HCM phenotypes. There was a Korean data which suggested a comprehensive genetic analysis of 212 patients. 82 were nuclear DNA, that is sarcomere associated genes, phenocopy genes and mitochondrial genes, and 37 mitochondrial DNA variants. This study revealed that pathogenic variants in sarcomere genes were more prevalent in non-apical HCM compared. So, it means very clearly somebody who has a sarcomeric mutation will have classic reverse type echo, this asymmetric septal hypertrophy. Septum will be 20 millimeter, posterior will be 12 millimeter. That is why we call it as ASH, asymmetric septal hypertrophy. Septum is more thicker than the posterior wall. That is more common in the regular cardiomyopathy compared to apical HCM. Interestingly, apical exhibited a higher frequency of troponin I mutation and lower frequency of myosin heavy chain 7. So, you have to understand MHC and myosin binding protein, both of them which are of the thick filaments, directly are proportional to a high thickness, high fibrosis, high risk of sudden death, rather than others which probably have a lower risk. Additionally, mitochondrial gene mtRNR2 was associated with apical hypertrophic. This has a distinct genetic profile, and apical HCM has got lesser mortality, lesser risk of sudden cardiac death. Japanese have got a higher incidence of apical HCMs, and Indians also quite a bit, but the risk of sudden death is much lesser. So, these findings highlight the heterogeneity of HCM and underscore the importance of considering both sarcomere and mitochondrial gene variants in understanding its phenotypic diversity. Impact of sarcomeric mutation on myocardium. So, several studies confirm that sarcomere mutations were related to increase in myocardial fibrosis. Kim investigated the effect of sarcomere mutation on myocardial mechanics and fibrosis pattern using cardiac MRI and echo. What did their study show? That sarcomeric mutations have lower circumference strain, that is low GLS, global circumference longitudinal strain is lesser, LV function is lesser, and they have got higher prevalence of mid-wall fibrosis. More the fibrosis, more the chance of arrhythmia, more the chance of sudden death. These alterations were independent of the extent of hypertrophy, indicating a direct impact of genetic mutation on. So that means a mutation can give you a clue. So I will probably do an echo, I will do a cardiac MR, I will do genetic analysis, try to risk stratify my patient and then take it further how to go about. This study provides further evidence of pivotal role of epigenetics for variable expressivity. Environment factors, hypertension, exertion, metabolic stress also significantly contributes to the disease variability. Now genotype positive but phenotype negative, high intensity athletic training or pressure overload may precipitate hypertrophy or arrhythmic events. That is what is important. So you only treat hypertension, let that athletic person reduce his physical activity, probably the hypertrophy is reversible, it can come down. So he is not the HCM category. Korean National Health Insurance tells you that association between body mass index and clinical HCM after the hazard ratio was almost 1.063. Metabolically unhealthy participant had higher incidence of HCM than healthy people regardless of obesity status. So somebody who is physically active, definitely the chances of developing HCM chances are lesser. So this means even when I am harboring a gene, my clinical physical activity and my lifestyle has got a great impact in what I have my presentation in the subsequent 2 or 3 decades. So this is also very, very relevant. So presentation of apparent can be sarcomere positive, sarcomere associated and monogenic hypertrophic cardiomyopathy. Syndromes, as I told you, they are mimics of hypertrophic cardiomyopathy, and polygenic, this is more of on the future role of polygenic risk scoring which we are talking about. And these are strong evidence for pathogenicity and limited evidence which I already have spoken about it, and percentage we have talked about. So if I talk about 2 or 3 genes, these 3 or 4 genes together, more than 90% of genotype positive cases will be picked up. And these are all the HCM mimics. Now there is a very interesting score that we call it as model for positive genetic assessment. If my age is below 45 years, 1 point. If I have septal thickness more than 20 millimeter, 1 more point. Family history of HCM, 1 point. Family history of sudden death, 1 point. Reverse curse hypertrophy, 1 point. So that is 5. But somebody who is having history of hypertension, it is -1. So range is from -1 to 5 points. Anybody who has 5 points, there is a 80% chance that my genetic testing will be positive. So history, if I have a family history of SCD, if I have a family history of hypertrophic cardiomyopathy, I am younger, below 45, I am having a septal thickness of 20 millimeters. If my score is 4, I have a 70% chance that genotypically I will probably have a positive result. Very simple. That is the reason that younger people, the chance of a genetic yield is higher. Somebody who crosses 40, 45, it's going to be a very difficult to pick up. So this is something which is very, very important. So now in a nutshell, genetics, these are established blue, diagnosis confirmation and family cascade screening. You need a genotype specialist to probably define how am I going to do the cascade screening. Means family tree needs to be done 3 generations before, 3 generations next, and then come to a conclusion what you should be doing. Polygenic risk score are still being established. Risk of phenotype development, tailored frequency in sarcomere negative families, interaction with all of them are questionable. We don't know a correlation. Targeted therapy, now we have genomics, or we have gene therapies, which I will come to it in later on, and risk stratification. Today, if somebody is genopositive but phenonegative, I cannot define the risk of SCD in that patient, and I cannot treat the patient only on genotypic positive factors. That is very important. And this is how it happens. when I have a protein and there is a no mutation, this is how the protein is developed, and this is a normal heart. The moment it goes, this is called as a truncated protein product, and this truncated protein activates the UPS pathway, which is going to be the ubiquitin pathway which we are talking about, and that directly results in hypertrophic cardiomyopathy. And this is also very important. How does history help me in genotype positive proband? If my family history is positive for HCM or SCD, reverse curve, female sex, septum more than 20, below 45, absence of hypertension, these are all positive signs that my genetic yield will be higher vis-a-vis all the others, the genetic yield may be lesser. This is exactly symptomatic. These are all the genes which have been identified. These purple and this is up to from here to here is the hypertrophic cardiomyopathy genes, sarcomere, filaments, M-band, Z-disc and sarcoplasmic reticulum and mitochondria. RAS, lysozyme, Z-disc are all related to symptomatic LVH but non-hypertrophic cardiomyopathy gene. So this is how we will be talking about pathogenic or likely pathogenic variants. All relatives, first degree relatives have to be tested. VUS, counsel on uncertainty, consider cascade with caution. Negative test, no further testing for relative, and genetic test not done, obviously we are not. So what you do is, in the families you need to look at ECG and 2D echo. Echo can be done, ECG can be done yearly, echo can be done once in 2 years or at the most once in 5 years, just to pick up whether this person is developing signs of hypertrophy on the 2D echo. Genotype guided therapy now we have. So current treatment remains largely phenotype based, like beta blockers, calcium channel blockers, disopyramide, septal reduction therapies like alcohol septal ablation, myectomy and ICD. These are all the medical and the non-medical surgeries. Mavacamtan is the newest drug, which is a myosin ATPase inhibitor, has shown promise in reducing hypercontractility. So in sarcomere genopositive, mavacamtan achieved primary composite with an odd ratio of 4.43 compared to 2.5 in the sarcomere genonegative in the explorer. So somebody who is genopositive, you straight away start mavacamtan, the chances of sudden cardiac death and the hypertrophy reduction will be much better than a genotype negative. Stronger and more sustained response has been observed in pathologic or non-pathogenic sarcomere, likely pathogenic sarcomere variants. Therefore, genetic testing may be useful in predicting therapeutic response and guiding personalized management strategies, including decisions like septal reduction surgery. Now, in gene therapy, there is a Cleveland Clinic data in 2023 targeting myosin binding protein C3 with an adeno-associated virus vector TN201. Preclinical data support reversal of disease through restoration of sarcomere. So at cellular level, you are restoring the protein expression so that the phenotypically the person doesn't develop. Gene editing technologies like CRISPR or Cas9 and RNA based are being explored for mutation correction or silencing, and these are the 4 varieties of gene therapy. One is called as the gene transfer. You send vectors and it transfers it into the DNA level. Splicing correction, which is called exon skipping. If this red one is the abnormal exon, you take it off. Instead of 1, 2, 3, 4, you have gene splicing 1, 2 and 4. This is mRNA without the mutant exon. The last is the repair which you can do, gene editing, which is the CRISPR and the Cas9 system, and the last is the silencing. At RNA interference you do. So there is a mutant mRNA degradation. These are different genetically based therapies. They are not yet active in clinical practice but going on. So maybe tomorrow genopositive people, before the child is born, you may start this, and maybe the person even though carries the gene but phenotypically doesn't express this problem. And these are all the therapies. I will skip for the audience or the crowd that we don't have much of, but remember, risk of complete heart block needs to be experienced, and this is ablation versus myectomy. This has got a more stronger benefit, so use it in the youngest people who have a large survival, healthy, long span, active, higher gradient, greater thickness and coexisting structural heart disease. Elderly, low gradient, sedentary, limited lifespan, you just go ahead and do an ablation which is there, which is called alcohol ablation. Now last few slides on genetic and family screening. This is the American Heart Association guidance. These are all class 1 recommendation in patients. Evaluation of family including a 3-generation family history is recommended. Genetic testing is beneficial. Atypical clinical presentation and another genetic condition is suspected, a workup which we do for the HCM phenocopies. Now we have genes for them, so you can do that. Genetic counseling by an expert geneticist has to be discussing. When performing genetic testing in proband, initial tier of genes tested should include genes with strong evidence. Strong evidence means 8 plus 3, 11 genes or 12 genes if I remember. First degree relative, both clinical screening and cascade genetic testing only when pathologic or likely pathologic variant has been identified in the proband. If it is abnormality with unknown significance, you don't do a genetic testing in their family members. Family with sudden unexplained death has occurred, post-mortem genetic testing also can be done. In patients who have undergone this, serial re-evaluation is identified to assess variant reclassification. Affected preconception and prenatal genetic as we already discussed. Not recommended HCM who have undergone genetic and found to have no pathogenic variant, they are only benign or like cascade testing is not useful. Ongoing clinical screening is not indicated in geno-negative relatives or VUS patients. And HCM, this I think we have already discussed. This is targeted gene testing, what you are going to do, we have already done that. Hypertrophic cardiomyopathy, this is what you see, apical aneurysm on the MRI, LV systolic dysfunction, unexplained syncope, massive LVH, family history sudden death, NSVT on Holter. This is for us as clinicians, we need to look at this. If my patient has this, he has a high risk of SCD and he probably can have sudden cardiac death. Now patient selection for ICD, this is very important, but each device costs you 6 lakh to 8 lakh rupees. If I have to put it in a young patient, I should have very strong evidence that this is going to benefit and it is going to be cost effective. So prior event, SCD, ventricular fibrillation or sustained VT, if it is present, straight away ICD recommended. If not, at least one of the following: family history, massive LVH, unexplained syncope, apical aneurysm, low EF. If it is yes, 5-year risk estimation, it's a 2A recommendation, so ICD is even reasonable in this group. No, if NSVT is present in children or adults, ICD can be recommended, but extensive gadolinium enhancement on MRI may be considered, but if it is not there, an ICD is not indicated. So this is very clear picture that look at all these features, do a ECG, echo, Holter, cardiac MRI, genotypic evaluation, phenotypic presentation, history of syncope, family history, all put together, calculate and then come and let us know how it is. So I'll skip all this. So in conclusion, most common genes identified in non-syndromic sarcomere is MYH7 for beta myosin heavy chain and myosin binding protein C3, cardiac troponin T and troponin I. These will account for more than 90% of your genotypic abnormalities that you see. They will account for 90% of genopositive. Finding of pathogenic or likely pathogenic mutation improves the diagnostic certainty, and you need to do family screening or cascade screening and evaluation in the family members with echo, ECG and genotypic evaluation, and accordingly all guidelines recommend genetic testing for HCM. And the future direction is the field is moving towards precision medicine, integrating genomics, transcriptomics and proteomics for personalized care. Longitudinal studies and registries are essential to validate geno-pheno association and assessing the long-term impact of the genetic interventions. Thank you very much for a patient hearing. I think we can take up any questions. Yeah.
So if we are normally when the patient comes, we the clinicians recommend go for the ECG, then for the 2D. Now, when you will be suggesting like monogenic inheritance needs to be checked for this particular patient?
So basically, if I have to evaluate the genetic, first of all, I should have a very strong feature. This patient has features of hypertrophic cardiomyopathy. So for that, that slide which tells you strong factors, a young patient who is below 45 years of age has got a ventricular septal thickness of more than 20 millimeters. He is the non-apical variant. Then I have a family history of sudden cardiac death or a family history of hypertrophic cardiomyopathy. These all features will make it very, very clear that if I do a genetic testing, my yield of a positive result is more than 60 to 70%. So those are the subsets whom on evaluation I will think he should go for a genetic testing.
So out of those 4 genes, which you will be recommending for the Indian population?
No, but now we have a genetic panel only. So it's not 4 genes, but I will have those 11 or 12 genes which will cover more than 90. That will be troponin C, troponin, myosin light chain, all will be a part. Now we have a panel.
Yeah.
So that they will take.
But only the known variants will be
Yes.
covered in that.
At that particular time, only the known. So 11 variants of the genes which account for more than 92 to 93% of the genotype issue will be picked up. So you'll pick up at least more than 93%. The other 5 to 7 are very rare. So they will not present at 30 years or 35 years or 40 years. They will present in childhood. So there you have to have a geneticist or a pediatrician also who should be able to pick this up in children. Now this fellow doesn't fit into, and then all of these genetic conditions have got separate clinical associations. Some acanthosis, some neuropathy, somebody has myopathy. So you have to look at not only the heart but all the skeletal features which should be. So you need to be an expert in genetic suspicion of a rare genetic disorder by which. So as a clinician, I may not be the only person, but I should definitely suspect. So ideally this should start from pediatrics. At adulthood, 18 years and onwards, I will see the classic garden variety of hypertrophic cardiomyopathy. Obstruction present, septum is thick, gradient is there, mitral regurg is there, and then I will probably take them up with medical treatment and all that. But those, what we are talking of, the rarer variants, they are more into the pediatric age groups which are there.
Okay. And then you can recommend the genetic counseling to those.
So that will be at that particular time we can think about, yes. Yeah.
So when should a person go to a doctor and think about genetic counseling or genetic testing of such diseases?
Ideally, a layman will not go unless and until you have a family history. Like in the past couple of generations, somebody has hypertrophic cardiomyopathy. So today, as compared to the robustness with which US does all these things, India even doesn't have specialty geneticist present. Today I have 1 person or 3 people who are across Pune. So if I have to call them, I have to take an appointment of them to come to my institute. Then I have to have the family sit with me like that. Maybe in US it's so systematic that from pediatrics onwards, you have a genetic expert. There is a genetic counselor who will look at the history and discuss all this. In India, we have not yet reached that stage. But yes, your answer to your question is, when should a layman go? Is only if you believe that I have a strong family history of sudden cardiac death. So definition of sudden cardiac death is a male below the age of 55 years in your family, maternal or paternal side, has had a death. Male below 55 and female below 65 have died suddenly, not accident related death, not confirmed myocardial infarction, post angioplasty, post bypass related death, not them. If such a kind of a thing is there and more than 2 family members are there, 2 or more, then you have a very strong family history. Then you go to a doctor, get your ECG and echo evaluated whether you have hypertrophic cardiomyopathy. If not, then the next common condition is coronary artery disease. I may be harboring a block. So I will get my 2D echo stress test. angiography, identify the block, treat the block. So that's not a familial condition. So it's very difficult for a layman to reach a doctor. It is only on routine screening. You go for a health checkup. Your echo shows muscle is thick. That's when the sequence, and that should be suspected by the doctor, and he should advise ki Baba, please go ahead and get your evaluation for HCM. That's how it is. But not routinely, it's very difficult for a layman to go, because in the general population, many people have just hypertension. But if you see hypertrophy in somebody who is below 45, that should click in your mind, this is hypertrophic cardiomyopathy, or any of the other variants that we are talking about. Professor, any points that you would want to add, I think, because you have been doing this over last 2.5 decades.
No, definitely. I think you have covered almost everything for this particular audience. Again, this genetic counseling is really the major part today, like to give awareness, impact and everything. Again, this is an opportunity for the audience to learn from here and then
Yes.
also.
Thank you. I know it was a very intense talk, but then there was no other way. I had to cover it because most of you are.
Each section can be a separate lecture.
I swear, absolutely. But I think I should thank that I learned what till now as a cardiologist I was not aware. With this talk, I think at least I have become much more aware. So thanks for the opportunity and thanks for inviting me.
I think you can turn off the light, this light. Oh, because it won't be bright. I have a lot of colors light. Should be fine. Yeah, perfect. Yeah. Okay, guys, good morning. I just need some energy in this room before we get started. So, my name is Sakthivel Sadayappan. I am from University of Arizona, Tucson. So, in the next 1 hour, I'm going to talk about, first, I'm going to talk about my background, who am I, where I come from, what I do, and then I'm going to focus a little bit about the secondary risk factors. I think yesterday Dr. Chandrakant Chavan, he indicated that 95% of the genetic carriers, they do not show disease. But if you have a secondary risk factors, diabetes, hypertension, anything else, how the genotype phenotype correlation can be presented. So that's a different, completely different area of kind of clinical aspect. So we are going to cover that today. And then at the end we have, I think at least I have some 10 quiz, just discussion, nothing to worry about any grades or anything. So these are my disclosures. Normally we do that when we give a talk in any places outside of the university, just to make sure that we don't have any conflict of interest. So I serve as a consultant for a number of companies, just to provide consultation on developing gene therapies to treat hypertrophic cardiomyopathy. So here is my big slide, one slide just to show about my academic journey. I received my PhD from Madurai Kamaraj University. This was in 1999. I also went to Germany as a DAD fellowship for 1 year on my PhD. I was the first one started the hypertrophic cardiomyopathy genetic screening. I will show you a couple of slides. Then also I spent 3 years as a postdoctoral fellow at the Max Planck Institute. This is the time I learned on clinical aspect, and then what should we do to identify the genetic risk factors. But again, it was not enough. As a basic scientist, I want to move on developing animal models, in vitro tools to understand the mechanism, and then also start developing therapies. So I went to Cincinnati Children's Hospital. This was in 2002. I was involved in developing animal models, studying cardiac physiology, doing echocardiogram, histology, gene profile, RNA-seq, etc., etc. So then I went to Loyola University of Chicago to start my independent lab. This was in 2009. I had my own team. I started training graduate students, postdoctoral fellows, etc. I was there for 7 years, and then I moved back to University of Cincinnati as a full professor. I was there for 8 years, again continue to do research and train all the lab members. And then a year ago, I went to University of Arizona. Now I am the head of the department, cellular and molecular medicine. Again, in my role, my job is to again promote, support the faculty, at the same time the department, and also I have to manage my research lab as well. So this is kind of Cincinnati in summertime. You can see very nice green like Pune right now, but in December you can see how much snow we get. So the move from here, from Madurai to Cincinnati, it's completely a different lifestyle. Nice meeting you. Now you can see this is my hometown here in South India, Vattanchatiram. This is Tucson, Arizona. Lot of mountains. It's a very nice landscape. You can see in this video, this is Arizona mountains. You can see when I was driving, I take this video. Big mountains, a lot of cactus. It's so beautiful. But in summertime, it's super hot, over 100 degrees. It's almost like this summer we had 108, very high temperature. In the middle of the day, you can't go outside. But it's a purely dry heat. It's not like heavy humidity or anything, so you can easily manage. Again, mentors matter. I'm standing here today, it's only because of all my mentors. They provided unconditional, always an excellent mentorship in my career. Again, I come from a very humble background from southern part of India. My parents never went to school. I managed every level in my career, and I'm here only because I got an excellent mentors in all my stages. So I always acknowledge them and thank them for their mentorship. Again, today I am committed to do the same. So this is my current lab members in Tucson, Arizona. I have a research assistant professor. I have 2 postdoctoral fellow. Kalyani, actually she was here in Pune University as a postdoctoral fellow. She received her PhD from IIT Madras. She is there now. And then I have graduate students, master students, and then lab managers. Again, my lab is heavily involved in studying muscle diseases, including hypertrophic cardiomyopathy. So again, my PhD work was on hypertrophic cardiomyopathy, but today we do a lot, just not the genome phenome analysis. We do animal models, we do molecular studies. We actually develop now iPSC-derived cardiomyocytes and then organoids from there, so we can test all the gene therapies, drug discoveries, etc. Number of fundings. And then in my postdoctoral time, I started expanding my research, understanding heart failure with preserved ejection fraction, inflammation, netosis, cardiotoxicity, etc. And now we also study skeletal diseases, specifically distal arthrogryposis. It's another genetic muscle disease, again focused on all genetic diseases. So these are my current research, in various aspects in the lab right now. So what are the experimental approach we do? Still I work on human clinical studies, specifically South Asians. We invite them to come to the hospital. We do echocardiogram, we do MRI, we collect blood sample, we do whole genome sequence. We also generate iPSC-derived cardiomyocytes from them so that we can study the phenotype-genotype correlation. We have mouse models, CRISPR-Cas9, induced, knock-in, knockout, transgenic, all type of mouse models we do. We also isolate single cells from the heart and then study their properties. You either skinned them, you remove all the membranes, then you can really study calcium-force relationship, or you can isolate freshly isolated adult cardiomyocytes, and then you can study their properties as well. So the story is that when I was a graduate student, I was given a project looking for biomarkers in heart failure. Pretty much this was 1995, those days. There was no internet, nothing. So my job was every day I go to the clinic, wait for the cardiologist. So they collect blood sample from heart failure patient, and then I take the sample, bring to the lab, separate the serum, I store that. Nothing else. This was going on for a number of months, because sometimes you go to the hospital, you wait there for a whole day, you get no sample. You just come back to the lab and there was no productivity. So after 6 months, I was able to collect only 25 samples, and really it was a boring project. Literally you do nothing. I mean, you can have all the samples, what can you do? You do ELISA, dot plot those days, not even ELISA, dot plots, and then you kind of look for up or down of these biomarkers. So I was not very excited. So that's the time this hypertrophic cardiomyopathy stories are coming up from other side of the world, from European countries, from USA. Genetic screening in Finland population, we identify novel mutation from German population. So I was able to see those kind of studies. Then I went to my mentor telling that, I'm going every day to the hospital. Why can't I collect blood sample from hypertrophic cardiomyopathy patient and then do genome sequencing or identify mutations? His answer was, it's a great idea, but we don't have even a PCR machine in the lab. So how can I do that? So then we kind of stopped that conversation. After a week, he came back again. He said, why don't you apply for DAD fellowship? So DAD fellowship is an exchange program. PhD students can go from India to Germany up to 1 year time. So then he recommended a lab, and that lab focused on candidate gene analysis. So if you have a genome DNA, they can do PCR, region by region by region, and then they can run the single strand conformation polymorphism, SSCP gel electrophoresis, and if they see a difference in the band migration, then they take it and then DNA sequence it. I applied for it. I sent a kind of 2-page proposal to the PI, that is Professor Hans-Peter Forsberg. Within 30 minutes, his response was, yes, we are interested, we will support you, you come, you spend time in my lab. So then I went back, so Dr. P.K. Joseph from Sri Chitra, he was my collaborator, and Sri Chitra is that in Trivandrum, they had a number of hypertrophic cardiomyopathy patient registry. So he identified 12 probands. Probands are the subject who come to the hospital with the symptom, and they called proband. So we collected 12 probands samples, and then 50 samples as a control from the campus. So I go to the lab by lab, convinced all the students and technicians to give blood sample. I was a phlebotomist. I had a very simple consent form. Those days, it was a very simple process. I extracted DNA sample. I put them all in one ice bucket. I went to the airport. Nobody stopped me those days. So I went to Germany. I spent 1 year time. My job was every day doing PCR. I started with the myosin heavy chain, and then I started working on myosin binding protein C. So these were the 2 top genes today we know have more number of mutations than any other sarcomeric genes. So we look for novel variants, and then we identify number of mutations. And that was my PhD work. And this is one of the families. After we identify the mutation, then I come back, then went back to the hospital, then track all the family members, collected blood sample. Now we can look only the identified gene variant. We don't need to do all the regions, so we can easily identify. In this family, there were 2 mutations, one in myosin heavy chain, one is in myosin binding protein C. Again, the subject who had both mutation was very severe, and even I think passed away after our work was published. And all this dark are symptomatic subject, and there were a couple of them are very mild symptoms, but pretty much very strong association of the genome-phenome correlation in this family. So later on, this myosin binding protein C, this is the 25 base pair deletion, was considered as a polymorphic variant in not just only in India, but also South Asian countries. So today we know that 4% that particular mutation is in Pakistan, 5% in India, 9% in Sri Lanka, and you can see the rest of the countries wherever South Asians are migrated. So this polymorphic variant, if you collect in this room samples and look for the variant, 6% of 100 people carry this variant, but the question is, do all of these carriers have disease? The answer is not, and that's what the secondary risk factors play a major role in developing the disease early on, or you don't have any disease. So we develop animal models. For example, this one, we overexpress the mutant gene. This is the normal mouse heart in 3 months. This is the 3 months of the mutant mouse heart. You can see the difference. It's really a defined hypertrophic cardiomyopathy phenotype. So we can study now using this mouse model at what time point you can really see the phenotype, what are the genes, what are the proteins that upregulated or downregulated, what are the post-translational modifications that happen in this developmental stages. So those are the opportunities if you have a model to study in the lab. If we can do the two-photon images. You can see that when you scan the heart, you can see the mutant heart, how big the ventricle wall thickness, etc. It's really mimicking the human hypertrophic cardiomyopathy condition. Okay, now I'm going to switch the gear a little bit on the secondary risk factors. Again, heart failure. Heart failure is a very big topic. When you look any type of heart disease, the end stage is the heart failure. Again, there are number of stages in developing heart failure, but again, I just put some of the numbers here in India. It's almost like 1.3 to 22 million people are affected with the heart failure. So again, you can see the reality is it's growing right now. It's not only in India but all over the world, like it is the number one killer. So if you can combine all type of cancer diseases, heart failure is the number one killer still all over the world. For example, patients, if they have the heart failure, 24% they die within a year of the diagnosis, and then 50% die within 5 years of diagnosis. It's very challenging, and again, we don't know what is really happening inside the body. We go to the hospital, only we know that there is a symptom, otherwise we stay on like ignoring such a small symptoms. So again, when you look at types of heart disease, there are so many types, and the cardiomyopathy, there are again several subtypes. Yesterday Dr. Chandrakant talked about it. Hypertrophic cardiomyopathy is one of the types. So I kind of going next to few slides to review hypertrophic cardiomyopathy. It's an autosomal dominant. Again, the sex different distribution is equal. In women it diagnosed less, and they have later onset. The estimate is 1 in 500. I think right now it's 1 in 3 to 400. And then you can see that there are symptoms, we talked about it, and then the differences in terms of the prevalence, genetic testing, and then the cardiovascular outcome by race or ethnicity, or even the economic status can also make a big contribution. So again, there are a lot of non-hypertrophic cardiomyopathy cases. They also cause left ventricular hypertrophy. Again, by definition, hypertrophic cardiomyopathy means purely genetic. You have to exclude any other secondary risk factors that involve in developing the disease. The summary is here. The major thing is diastolic dysfunction. The patients, they have improper relaxation. That's one of the major criteria for hypertrophic cardiomyopathy. Diagnosis, we do all this kind of thing: genetic testing, echocardiogram, electrocardiogram, and then exercise stress test, and then MRI. Again, we covered yesterday. And then the treatment, again we talked about beta blocker, calcium channel blocker. Right now we have the drug called mavacamten. It's only one approved by FDA, available today in the clinic to treat specifically called hypertrophic cardiomyopathy. And then all the surgical procedures. The global distribution, almost 125 countries. Documented, published work on defining hypertrophic cardiomyopathy, and actually, the prediction is that it's more higher than previously proposed. The more genetic testing you do, you identify more gene variants, then also the disease outcome. So the major challenge why we have to study the secondary risk factor is the clinical spectrum of the disease is highly heterogeneous. What does that mean? It means that within a family, if you take one gene mutation, the disease outcome can be varied between individuals. Some subject they can develop very mild, moderate or very severe disease. The penetrance, some people can start with very early on in their life or the middle or later in stage. And then also the phenotype, the thickness, the function, developing arrhythmias, histology, fibrosis, this all can be varied between individuals. And the reason why, we don't know. So that's what we are trying to understand. What are the factors that involve in this variability? For example, you have a normal heart and then you develop hypertrophic cardiomyopathy. The question is, if you have modifier genes or secondary risk factors such as hypertension, diabetic, etc., or the lifestyle choices, the outcome can be very significantly worsen phenotype. They can develop heart failure by having these secondary risk factors. Instead of one genetic variant, if you have two different genetic variant in two different genes, that can cause severe phenotype than just one gene variant. So primary versus secondary risk factors, genetic mutations versus modifiers. So sarcomeric genes are the primary genes, and if we have mutation in sarcomeric genes could cause hypertrophic cardiomyopathy, but the modifier gene mutations can add into that. And then why secondary factors matter? It really matters because that differentiate the variability or the heterogeneous in the disease development. And then the morbidity, mortality, sudden cardiac death, they all related to the secondary risk factors. There are a lot of influences. For example, as we know from yesterday's talk, severe exercise, for example, right? Athletes, they pass on the field because they go through this secondary risk factor. So today we are going to focus on understand key secondary risk factors and then their mechanistic role and then clinical management implication. So if you are a clinician and if you are looking for a patient that they have these secondary risk factors plus hypertrophic cardiomyopathy, how do you manage them? And there are also non-modifiable secondary risk factors like aging, for example, that can also relate to the fibrosis and diastolic dysfunction, sex differences. Again, we talked about that one, severity in males, and then females are much more protected than males. Ethnicity, there are differences in racial. So again, in some population, they show more prone than any other population. For example, South Asians, we have 3 to 4 times higher risk for heart disease than any other population. It's well documented. And then family history, again penetrance, the phenotypic variability, this all comes under the family history. So modifiable secondary risk factors, again I kind of covered. So smoking can lead to endothelial dysfunction. At the end, you develop myocardial infarction. So if you have a hypertrophic cardiomyopathy, if you smoke too much, you develop myocardial infarction. Comparing to a non-smoker, non-myocardial infarction, the risk is very high for the patients with the smoking habit. Physical inactivity, again lack of exercise, lack of kind of activities can also cause into this. Then alcohol, substance use. So you can see all these secondary risk factors are directly associated with the development of or the severity of the phenotype development. Lifestyle and environmental triggers, again exercise can also cause stress. People, for example, the broken heart syndrome, it's one of the syndrome that develop very quick heart failure. If you are really stressed out, unable to sleep, constant thinking, those all can cause into the stress and then related to the developing a severe phenotype. Dietary factors, people take a lot of salt or obesity, they also link to the severity of hypertrophic cardiomyopathy. And then lastly, environmental stresses, altitude or heat, pollutants, this all could also involve in the severity of hypertrophic cardiomyopathy phenotype development. So this is the question I always talk about it, right? When you say, I have the genetic, is it enough to cause the disease? Or the Hyderabad biryani, or the jalebi we are eating, or the laddu we are eating, right? This all can add into the disease development. What do you think? Yes? 100% yes. So, when we talk about Indians or South Asians, there is a very specific phenotype. Do you know what I am talking about? So South Asian countries are Pakistan, India, Nepal, Bhutan, Bangladesh, Sri Lanka, and then a lot of small other countries. We represent 1.8 billion. Indians almost now 1.5 billion. Population is very, very heavy. And we have 4 times more likely to develop cardiovascular disease than any other population, any other ethnics. The common misconception is obesity considered a sign of wealth and good living? What do you think? Yes or no? No, right? But when I was a child, this was the case. You have to have a belly. You have to have like more muscle. Then they think like you are so wealthy and happy guy. Sometime now, even now, when I go to my village, people say, Sakti, what happened to you? You look so thin. Is your wife is feeding you or not? This is very common comment they always give about it. Secondly, are Indians generally not overweight? Yes or no? They have bellies, right? If you look at any of the people after 40 years, 50 years, they have the obese. Is a vegetarian diet considered healthier? I know in this room some of my colleagues are pure vegetarians, but they think they are very healthy. Again, the answer is no. The next one is the lifestyle choice. So we don't normally do exercise. That's kind of our culture, and we eat a lot of carb, so much rice. When you go to any festival, big banana leaf, so much rice and little sambar and even some vegetables. That's it pretty much, so but we don't do exercise. Those days when we were like 50 years, 100 years, it's a different story. And here you can see, today people stop cooking. Everybody is going to the restaurant and eating all junk. You go to the hospital, you ask for a doctor, they give you the medicine. You wanted to know what exactly this medicine is, how it works, what are the secondary risk factors. But when you go to a restaurant, when you eat, you don't ask all those questions. You only look for the taste. And they put anything to make you happy. And down the road, there is a big consequences. And again, we eat a lot of this fried food, so much calories. So here some of the calorie calculations. I don't know whether you know that. If you eat one piece of laddu, 185 calories, one piece of jalebi, 150, rasamalai, my favorite one, 180 calories, one piece. And gulab jam, again 192 calories. Think about this. You are the next generation. When you are with your parents, with your grandparents, with your families and friends, you need to remind them. People eat so much and it's not good, without doing exercise. And now even worse, we are now exposed to the Western diet, all this burgers, fries, pizza, and without doing any exercise. So again, these are the things when you talk about any heart disease should come into your mind. Secondary risk factors, a quality of life, moderate diet, moderate exercise, and good sleep. You should sleep better. 6 hours essential. Completely uninterrupted sleep is so important. Mental health, it's also so important. How happy you are. These 4 things are directly linked to one another. If you don't sleep well, you don't eat well. Or if you are mentally ill, you eat too much. So there are consequences in these 4 factors. So you have to really pay attention down the road or promote this kind of awareness to our community. Hypertension is very common in India, for example. So this is the one in the States, they always talk about know your numbers. This is the phrase they say, know your numbers. So just to measure your blood pressure, you don't need to go to the clinic. You buy simple the blood pressure measurement thing, you can do it. It's a very simple, non-invasive procedure, and I don't know how many elderly people, they do it routinely at their home. So less than 100, this is the sugar level, and then the blood pressure should be 120/80. Systolic, diastolic pressure should be there, and then cholesterol level. These 3 factors are really important to have a healthy lifestyle. Again, they are linked to one another. If you have higher sugar level in the blood, causes diabetic, and that result in changes in the rest of these 2 parameters. So these are very simple diagnostic tests these days. If you are 40 and above, you should do it every year. Cholesterol check, blood pressure at least every 3 months, and blood sugar at least every year. You have to know these numbers on record so that you can compare how you do it down the road. Here body mass index, again you have to have at least 1.8 to 2.9. It's very ideal to have these numbers. When you define hypertension, again the normal is 120/80, and the elevated is 130 or 129 to greater than 80. Stage 1, it goes to 139 to almost 90, and then defined hypertension stage 2 is 140/90. Again, it's very, very common in India. I have seen so many of my friends and family, they do have this higher hypertension, and they don't pay attention. They just don't take the medication on a regular basis, and they don't understand the cause for this hypertension. Instead of taking medicine, why can't they just walk every day 30 minutes, do some exercise, change your diet, change your lifestyle? That can easily change everything. That's what the Hypertrophic Cardiomyopathy Clinic also today recommending. Because you have hypercontraction, your heart is always hyperfunction, change your lifestyle. Don't run to go to your job, not eating your breakfast, not doing exercise, lot of things, So if you see there is something like disease coming, you need to change your lifestyle for sure. Pathophysiology and mechanism. So again, we are going to really deep dive into this area in the next couple of lectures. We will talk about sarcomere dysfunction and how secondary risk factors can cause imbalance and cause severe phenotype, fibrosis and remodeling pathway. Again, hypertrophy plus secondary risk factors can aggravate, increases the fibrosis level. Then energetic imbalance, again diabetic people, for example obesity people, they have the energy imbalance, right? And the heart utilize maximum the fatty acid as an energy source and then switch to the glucose one. So when we don't have a proper balance, then that changes the entire cardiac function. Then the molecular pathways. So again, because of all these changes, we can have increased inflammation or the secondary oxidative stress. This all can cause mitochondrial dysfunction and then down the road a severe phenotype. Clinical implications, again in the States they have a clear risk stratification models. They integrate secondary risk factors, the scoring system, and then personalized therapy. So again, targeting modifiable risk plus genetics. You have to really work on both, not just treating hypertrophic cardiomyopathy and then eating burgers every day or pizzas every day. That's not going to work. And then preventive cardiology in hypertrophic cardiomyopathy, mostly lifestyle interventions. So again, you have to have specialized doctors to talk about what are the lifestyle you should choose and then how that can modify the development of the disease. And then the clinical decision making, that's between the cardiologist and the patient. So the key take home message is that secondary risk factors 100% influences the development of hypertrophic cardiomyopathy for sure. Modifiable risk, that's an opportunity for intervention, and then if you talk about hypertrophic cardiomyopathy, you have to focus on the genetics, diagnosis, biomarkers and lifestyle changes in precision medicine. So these are kind of future direction. Today AI, machine learning are now come up in terms of combining all type of this kind of risk factors, genetic and the all the biomarker or echocardiogram, ECG, all the parameters put together and then give you what kind of treatment option you should go down the road. So all those kind of things are coming up right now. So now we are going to a Q&A and discussion part. Before I jump into it, I want to hear from you any questions, comments before we go into the Q&A and some cues for you. Thank you. Yes. We still have time.
Hello, sir. How the progesterone prevent this disease? Because you mentioned that because of progesterone level, female are less sufferer.
So, number of cardiac genes in the promoter regions, they do have a transcription binding site. So, the hormones binding sites are in those genes. Either they can activate, they can inactivate based on the hormone level. And that is really kind of the cardioprotective effect seen by the hormone.
But testosterone is responsible for more muscularity.
Right.
We can contradict testosterone effect and
So again, related to the disease development, based on the age, the level of hormones, so number of factors again involved in this one. So it's not just a level and then disease. There are a number of factors they look into it right now. Yeah, but definitely it play a role in the disease outcome. Yeah, definitely.
Really nice presentation. I just
Yes, so infection, like any virus infection, COVID for example, they all possibly, there is a virus called Coxsackie B virus, I don't know, that can directly target cardiac muscle, and so if you expose those things, definitely can aggravate or cause severe disease outcome. I am not sure about the vaccination right now. There is no study, for example, COVID vaccine, how that influences the development of or severity of hypertrophic cardiomyopathy. Specifically, we don't know. It's possible. Anything you affect, so remember, you born with a mutation, right? And then you grow, right? Based on how you grow in your life, right? If you are a calm guy, like wake up in the morning, whenever you want to, have a bed coffee, slow day, your symptom development is very slow because you always been very calm, But if you are a very active guy, run, rush, stress level is high, you develop faster. So there is a direct link between level of stress, the disease development. So everything, that's what they say, change your lifestyle, that can give you better disease management. But you can't sleep longer, those days are gone.
Hello.
Yeah.
So nice presentation. Yeah. So my question is a very general question. So I hear a lot of people that they develop more cardiac disease, cardiovascular disease after this COVID onset, or maybe they recovered after this COVID diseases. So do you know by chance whether there is any molecular connection between these two or any kind of, do they really exist or it's just by chance it's happening?
So for sure, I think the COVID virus can infect cardiomyocytes, right? They can target, I think there is a receptor, that receptor COVID protein can bind in cardiomyocytes that can also infect. But the inflammation, all the risk can go and target heart muscle, The more inflammation you start developing, then you develop more fibrosis, sudden cardiac, that's called the arrhythmias and then sudden cardiac death. You probably see a lot of people died suddenly. They don't have myocardial infarction or heart attack, but they had sudden cardiac arrest, because of cardiac arrhythmias and factors like those kind of things.
It was not something which was affected by the virus, it was the secondary effects.
Yes, secondary effect.
Mm-hmm.
Yeah. Yeah. But again, I want to disclose, I am not a clinician, I am a basic scientist.
It's a general question.
Don't
sir.
take my clinical advice as a...
Okay. Sir, I am Dr. Padmini Doipude, sir. I have done my PhD on diabetes itself, comparative drug analysis. So, I just want to ask you that how many percentage of diabetic people may be affected through SCM?
So there is no real number or percentage. Right now, actually, we started a center study in Chennai. screening all the diabetic people for genetic variants in sarcomeric genes that could cause hypertrophic cardiomyopathy, and comparing them diabetic versus non-diabetic and the disease development.
Okay. So,
but we don't have a specific.
You mentioned that type 2 diabetic people are more prone towards SCM.
Yeah.
And what about type 1 juvenile?
So again, anything that related to cardiac abnormality, changes cardiac function. So again, remember, you have a sarcomeric gene that develop hypertrophic cardiomyopathy.
Yes,
Now
yes.
you are adding the additional risk factors to the heart that could cause severe, more early disease development for sure.
Okay.
And so when you treat, so you have to treat both. You can't just treat only one and then leave the other one. So that's why they try to look like today, when you visit a clinic, they give you the questionnaire. What else you have? Family history you have, but do you have hypertension? Do you have diabetic? Do you have this? They ask all the information.
Okay. Thank you, sir. Thank you very much.
Sir, Indian parents especially, before becoming a parents, they dream of their kids to be something like, sports person or other athletes. So in this case, is it the right time that, parents screen their kids before pushing them into the sports? And is there any, any clinical setups where we can do this kind of, sarcomeric gene mutation screening? That's one thing. And second thing, is there any direct correlation between the performance of athlete with sarcomeric and the athlete with sarcomeric? Is there any correlation between, the level of their performance?
Yeah, so the first one, so it's not in India but in the States. There are a lot of non-profit organizations. They go to high school, 11th grade, 12th grade. They start doing ECG. All the students get ECG at 11th grade because the students, they are very young. If they have any abnormal cardiac function, they could predict early on, right? That's one thing they started doing it. But there is no real like a program. If you are getting into the sports, there is a standard, gene testing for any games or any sports. There is no such a one. But if you are a parent, you want to know, you can do it, on your own cost. It's a prevention, so the insurance will not cover it, but you can pay out of the pocket to do like a gene panel. So they have, the Illumina has specific panels for hypertrophic cardiomyopathy, sarcomeric genes, or they have all the genes. So there are various panels. You can order them. You can send the blood in a tube and you get the result. So those are readily available. But again, that's a proactive these days, getting a lot of awareness. People have started doing it by themselves. But in India, I don't think so. So that's one thing. Second thing is, if you have a mutation, if you have the disease, how is your performance as a sports person? I think in hypertrophic cardiomyopathy, again, I don't have any experience, but again, there should be some poor performance. They can't run faster, they can't perform faster because they get easily shortness, breath shortness they develop. And so I don't think they can perform very well. But they push hard because they feel like underperforming maybe, then that causes a sudden cardiac death or arrest.
Thank you, sir.
Sir, you have more experience in genetic screening, right, about this disease. So whether mutated gene is more expressed or they are less expressed in this condition?
Yeah, so if you do the genetic screening, you identify either missense mutation, right? And if the missense mutation is pathogenic, that can cause either gain of function or loss of function based on where the amino acid mutation is localized, right? So that's one aspect. If you take myosin heavy chain, 90% of the mutations cause gain of function, resulting hypercontraction. Like they have more active myosin interaction. I think Princess can explain the structure dynamic. He's the expert in studying the ultrastructure of sarcomeres. So if they have gain of function, they interact more with actin, so they develop more cross bridges, and so meaning that more hypercontraction. Loss of function develop slow function and dilated cardiomyopathy. So this is one aspect. But if you have a truncation, you have a stop codon in the middle of the protein and you don't have the full length protein, what causes, right? Number one, these proteins can be, the mutant protein can be a poison peptide because it's non-functional, expresses and stick to randomly some area in the sarcomere and cause problem. That's one aspect. If you have a very strong, good ubiquitinated proteasomal system, you remove this mutant protein so fast. So now you are living with only one allele, right? Because that's a wild type allele, and the amount of protein that generate from the wild type allele is not enough. That called haploinsufficiency. Again, down the road, based on the gene and protein and either gain of function or loss of function. So again you go into classification.
Sir, in gain of function, is there any possibility to create the vaccine by using antisense technology like this?
So people are now doing all, instead of vaccine, they look for antisense oligos. These antisense oligos can go and remove the mutant mRNA, or the CRISPR-Cas9 that can go, binds to the promoter, activate the promoter activity, so you have more expression. So those are the things that are going on right now. Yeah. Great questions. There is one in the back. Will come.
The chronic kidney diseases, are they one of the factors of hypertrophy?
Yes, again, anything you have in the body other than normal function, they all could lead to severe disease development. If you have, these are called secondary risk factors, right? Having a kidney disease, then you are the blood, all the salt conditions, everything, it changes, right? That causes problem with the cardiac function as well. So 100% yes.
So I would like to add into this. So regarding kidney disease.
Yes.
So you, as a zoology students, you studied the function and structure of the kidney, and you know that kidney has important function. Kidney also controls the function of heart.
It's working.
Yes, it also controls the function of heart. How it, you might have heard, renin angiotensin aldosterone system. Through that system, it controls the function of hearts, and it maintains the blood pressure, and also it has an important role in maintaining the vascular tone. So therefore, if there is any problem in the kidney, heart is affected. And in this regard also, I will also like to add one more thing, the question which was asked by Dr. Shinde. So you asked about the progesterone. So progesterone has important role in maintaining the vascular tone and the blood pressure. So through these various long cascades of mechanism of action, progesterone activates certain enzymes which are responsible for the vasodilations, and therefore the blood pressure is maintained and the vascular tone is also maintained. Okay, thank you.
Sir, you said that people with South Asian people have higher susceptibility to this disease. So my question is, when South Indian people migrate to countries with completely different climates, like Canada, Netherlands, or Ireland, countries like those where altitude, temperature, humidity is completely different, do those people retain their susceptibility to this disease, or is there any alteration in that?
Yeah, so far studies from the States or even from Canada, I don't think any studies on the altitude, but definitely for the exposing to Western diet, because we go to States and there we are exposed to McDonald's, like burgers and fries and pizzas. This is completely a Western diet. Our genes are not inbuilt to process this, and we have very small, arteries. So again, we have a high. This could all lead to increasing cholesterol or hypertension, diabetic, obese. So we have, even in the States, we have a higher cardiovascular diseases than any other population.
And what exactly are the factors which make South Asian people more susceptible to this disease?
That's $1 million question. Definitely the lifestyle choice, one for sure. But again, we have a lot of infectious diseases. I think compared to maybe 100 years now, it's much better. Treatment option, for example, many people, the lack of awareness is one of the major thing that we have increased in cardiovascular diseases. What I mean that is, if I have a heart attack, I don't know what does that mean. First of all, I'm not aware of the symptoms of the heart attack, right? So I assume it's a heartburn, and I tell my family, we can go to the hospital tomorrow or whenever I'm free from my work. That's not good, because they die. The more delay you have before you go to the hospital, you have very severe outcome, within 90 minutes. So in the States, they call 90 minutes window. You call 911, within 90 minutes, you are in the cath lab going through the procedure. That's what they do. If they do that, 100% it's protected. You back to normal. The more delay you go, you have more damages in the heart. So lack of awareness, we need to promote, we need to tell people what types of heart disease, how to prevent, what are the lifestyle. These are the awareness we need to create within our community. I think that's one thing. Without, again, I tell you, right now people don't want to cook. They are going out, and we don't know what the restaurants are giving you. When you go to the clinic, you talk to the doctors, you ask all the questions. But when you go to the restaurant, you don't ask any question. You just buy and eat. It's all not so good. So I think now I see that ICMR is now introducing the calorie labeling in most of the food. I just hear that last month they're starting it. It's kind of improvement. If you know the calories, then you probably aware how much you are eating, what's the consequences. A lot of factors, yeah.
So I just wanted to ask, if a person is genopositive, can he avoid the SCM by just controlling the secondary risk factors? Is it possible?
It is possible, but not 100% avoiding. You can minimize the development of the severe phenotype early on. So the aging itself, there is a direct correlation between the disease development and the severity. But if you have a very good lifestyle, you can manage the disease so well. So that's what it is. Thank you.
Any more questions?
So we are getting time tea break and then okay, tea break and then we come back for this.
So right now we are going for a tea break, and thank you so much, sir, for this wonderful talk. And we will meet again after 30 minutes at sharp 11:30 a.m. So thank you.
After that, after tea break, there would be a tutorial session, so be prepared for that. And don't worry about this, it's very simple, it would be very simple. Don't worry, be calm and happily we will complete our course. Don't worry about the tutorials. Okay? Thank you.
Where should we go for tea? Is in this room or the same place?
Yeah, we will be here also.
Here. So the tea will come here.
नेटवर्कचा इश्यू होतोय हां असं ठेवा एकच आहे ना का दोन्ही आहेत हे दुसरंच आहे ना हां एक मिनटं ते वायर घ्या जरा थोडीशी हां थांबा ओके पीछे जाके बात
हेलो हेलो हेलो
हेलो
हेलो हेलो